Hello and welcome to MEDSimplified, a channel where we make biology and medicine simple. In this video, we're going to learn about the structure and function of a nephron. Nephron is the basic functional unit of the kidneys that consists of a renal corpuscle and the tubule attached to it. The word nephron is derived from the Greek word nephros, meaning kidney. Each kidney contains approximately 1 million nephrons, which perform several important functions of our body, including waste excretion, maintaining the body's pH, regulation of blood pressure, and several other functions. So let's go through the structure and function of a nephron and learn more in detail about the different parts of nephron and the role of renal tubule in urine formation and excretion process. Let's first start with the basic structure of a nephron. So nephrons are microscopic structures located in the kidney, and kidney has two important gross regions, an outer cortex and an inner medulla. And if you look at this nephron, you can see that a part of this nephron is present in the cortex of the kidney and part of this nephron is placed in the medulla of the kidney. This is an important point to remember as it plays a significant role in the formation of urine that we will discuss later in this video. So the nephron is a tube-like structure whose length varies from 35 to 55 millimeters, and the structure of nephron can be basically divided into two major portions: the Bowman's capsule and the renal tubule. At one end, the tube is closed, folded, and expanded into a cup-like structure which is known as the Bowman's capsule. Now, the Bowman's capsule was discovered by this guy, Sir William Bowman, at the age of 25. He was an English surgeon and histologist who is also credited with the publications of two historical books, The Physiological Anatomy and Physiology of Man, Encyclopedia of Anatomy and Physiology. So coming back to the nephron, we discussed about the cup-shaped structure at one end, which is known as the Bowman's capsule. The space inside the Bowman's capsule is known as the Bowman's space. The Bowman's capsule encloses a cluster of blood vessels which is known as the glomerulus. The word glomerulus is derived from the Latin word glomer, meaning a ball of thread. So you can see why this is known as a glomerulus. The blood vessel which brings the blood inside the glomerulus is known as the afferent vessel. And the blood vessel taking the blood out of the glomerulus is known as the efferent vessel. Remember, A comes before E, so afferent brings the blood in and efferent takes the blood out. The Bowman's capsule and glomerulus together constitute the renal corpuscle. Okay, so this was about one end of the nephron. The other end is known as the renal tubule. Now, the word tubule just means a small tube, and as you can see, the renal tubule is a long and folded tube-like structure that emerges from the Bowman's capsule at one end and it ends into the duct system of the kidney. The tubule is divided into three main parts based on its function. The proximal convoluted tubule, which is this part of the tubule shown in the light blue color. Then we have the loop of Henle, which is shown in dark blue color. And finally, we have the distal convoluted tubule, which is this part of tubule here shown in orange color. The distal convoluted tubule in turn ends into the collecting duct. So the first part of the tubule is known as the proximal convoluted tubule. Proximal because it is close to the Bowman's capsule, and convoluted meaning it is a highly folded structure that helps to increase the overall length of the tubule in a small space. The proximal tubule is located in the cortex of the kidney. The proximal tubule leads to the loop of Henle, which is the second main part of the renal tubule. This region forms a loop that is located in the renal medulla. The loop has a descending limb and an ascending limb. The ascending limb of loop of Henle leads into the distal convoluted tubule. Distal meaning away, since it is located far from the Bowman's capsule as compared to the proximal tubule, and convoluted, since it is also a folded structure to increase the overall length of the tubule. The distal convoluted tubule drains into the collecting duct, which also receives input from many other nephrons in the region. The collecting duct drains into the larger collecting ducts and ultimately into the renal pelvis, which is connected to the ureter. The ureter drains the kidney to the bladder. The distal convoluted tubule is also located in the cortex of the kidney. Now, depending upon the length of the loop of Henle, we have two types of nephrons in the kidney. One type of nephron that have short loops of Henle, and these are known as the short cortical nephrons. Cortical because a major portion of these nephrons is located in the cortex of the kidney. These are short and comprise about 80% of the total nephrons. The other type of nephrons that we have in our kidney are known as the juxtamedullary nephrons, which have long loops of Henle that are mostly present in the medulla of the kidney. These two types of nephrons can concentrate urine up to different osmolarities that we will discuss when we talk about the function of nephron. These are 20% in number. Now, along the whole length of the nephron runs this network of small blood vessels known as capillaries that brings in impure blood containing many waste metabolic products and takes out pure blood that has been filtered by the nephron. The impure blood or the arterial blood comes into the kidney via the renal artery, and the pure blood, which has been filtered of all the metabolic and other waste products, goes back to the body via the renal vein. So this was about the structure of nephron. Next, let's have a look at its basic function. So the nephron uses four main mechanisms to convert blood into urine, which are filtration, reabsorption, secretion, and excretion. Now, I have seen most of the biology teachers here on YouTube, and what they do is they just explain what is filtration, what is reabsorption, what is secretion, and job done. This is how urine is formed. Well, not quite right. See, in this video, we will understand the mechanism of urine formation in a better way. We will look at how the blood is filtered at the level of renal corpuscle, and then we will understand how the urine moves forward in the renal tubule and how different mechanisms like reabsorption and secretion apply in different parts of the renal tubule. So the blood enters the kidney via the renal artery. This is a large artery and a branch of aorta. After entering the kidney, the renal artery gets divided into many small divisions until it reaches the smallest one that enters the nephron. This is the afferent vessel that brings the blood into the glomerulus, and this is the blood vessel that takes the blood out of glomerulus, and this is known as the efferent blood vessel. In between these afferent and efferent blood vessels is a jumbled up mass of capillaries that is known as the glomerulus. Like we discussed earlier, glomerulus is derived from the word glomer, which means a ball of thread. The glomerulus is a tuft of blood vessels that is located inside the Bowman's capsule. This tuft of vessels greatly increases the surface area for filtration that comes in contact with the Bowman's capsule. And this is where the first mechanism of urine formation takes place, that is known as the filtration. And to understand the filtration at this site, you have to know a little bit about the glomerular filtration barrier. So if you take this part of the renal corpuscle and look at its microscopic view, it would look something like this. This is the wall of the blood vessel. Outside this is the basement membrane of the glomerulus, which is in contact with the wall of the Bowman's capsule. The wall of the Bowman's capsule contains special type of cells which are known as podocytes. And between these podocytes we have small gaps, as you can see, and these are known as the filtration slits. Together, these three layers form the glomerular filtration barrier that acts like a filter paper. So we have blood coming in this blood vessel that gets filtered through this glomerular filtration barrier to form the impure plasma that is passed down into the renal tubule. The glomerular blood pressure provides the driving force for water and solutes to be filtered out of the blood plasma and into the interior of the Bowman's capsule called the Bowman's space. Only about 1/5 of the plasma is filtered in the glomerulus and the rest passes into the efferent arteriole. The things that do not get filtered are things like red blood cells or larger molecules like proteins. Also, one important thing you should note here is the difference in the diameter of the afferent and the efferent blood vessel. As you can see, the efferent vessel is of smaller diameter as compared to the afferent vessel, which means that the blood is coming in through a vessel of larger diameter and leaving through a vessel of smaller diameter. This increases the pressure inside the glomerulus, which provides a driving force which makes this filtration possible. The kidneys can adjust the rate of glomerular filtration by adjusting the pressure inside the glomerulus, and this is all possible because the diameter of afferent and efferent blood vessels can be adjusted. The plasma that gets filtered into the Bowman space is known as the glomerular filtrate. The glomerular filtrate next moves into the renal tubule, where it is further processed to form urine. This further processing is very important as the initial glomerular fluid that is filtered into the Bowman space contains a lot of things that shouldn't be excreted out with urine, like sodium chloride, glucose, amino acids, and excess water. The first part of renal tubule is the proximal convoluted tubule, where approximately 2/3 of the filtered salts and water, almost all glucose and amino acids are reabsorbed. We don't want to lose glucose. That's hard-earned stuff that we ate that was good for energy. We don't want to lose necessarily as much sodium chloride as well. This is achieved by the method of reabsorption. The whole length of the renal tubule is surrounded with blood vessels which are known as the peritubular capillaries. Whatever that gets reabsorbed is then passed back into the blood circulation. Now, this is known as reabsorption and not absorption because the same nutrients have been absorbed once from your gut into your blood by the similar mechanism. The wall of the renal tubule has many types of protein channels that reabsorb some of the important things that shouldn't be excreted with urine. Now, about 70% of sodium chloride gets reabsorbed in the proximal convoluted tubule, and same 70% water gets reabsorbed here. And more than 95% of glucose and amino acids get reabsorbed in the proximal convoluted tubule. Next, the filtrate passes into the loop of Henle. The loop of Henle has some role in reabsorption of important salts, like about 25% of sodium gets reabsorbed here. Similarly, 25% of calcium, 20% of potassium, and about 60% of magnesium gets reabsorbed in the loop of Henle. But the loop of Henle has a major role in the concentration of urine. And this is a complex topic and usually taught at the level of med school physiology, but I'll try to simplify this as much as possible. So pay close attention. So here is our section of kidney, and you can see how the structure of kidney is divided into an outer cortex and an inner medulla. The inner medulla of the kidney is a very important region as this contains higher concentration of salts as compared to the cortex of the kidney. This higher concentration of salts in the medulla is made possible by the loop of Henle of nephrons. So here is our nephron inside the kidney, and you can see how this loop of Henle is present inside the medulla of the kidney, and the rest of the nephron is present up in the outer cortex. The loop of Henle has a descending limb and an ascending limb. So if you look at the ascending limb of loop of Henle, it contains special ion channels that pump out ions like sodium and chloride, thus making the renal medulla more salty or hypertonic. The ascending limb, however, does not allow water to move out, as it has no channels in its wall that allow the water to move out. These types of channels that allow water to move out are known as the aquaporins. And these type of channels are actually present in the descending limb of loop of Henle that makes it freely permeable to water. So now this is all salty, and the water will move out of the descending limb into the renal medulla and then eventually into the blood vessels and gets reabsorbed. And this is the reason why loop of Henle is so long. that is to give time for this water to secrete out. And that is why it dips nice and pretty far down into this salty portion, which is the renal medulla. So this is the major mechanism of gaining back a lot of water that gets filtered up at the level of renal corpuscle. So after the loop of Henle, the water then passes into the distal convoluted tubule, which is this part of the renal tubule. And here we have more reabsorption of ions like calcium and sodium. We are just reabsorbing more things that we didn't want to lose in the first place. There's a lot of things we could talk about what gets reabsorbed here, but this is just the overview. In distal convoluted tubule, the final mechanism of urine formation also takes place, that is known as secretion. You can view secretion as the opposite of reabsorption. In secretion, the waste ions and hydrogen ions pass from the capillaries into the renal tubule. This is the final step in the formation of urine, during which walls of the tubule actively remove waste substances which are harmful to the body from the blood that have escaped filtration. The secreted ions combine with the remaining filtrate and become urine. The major substances secreted into the distal convoluted tubule include hydrogen ions, potassium ions, and ammonium ions. The mechanism of secretion also takes place in the proximal convoluted tubule, but to a lesser extent. And some important stuff that gets secreted into the proximal convoluted tubule include substances like urea, uric acid, creatine, ammonium ions, and some other drugs. Then at the end right here, we have our filtrate that has been processed. A lot of water has been taken out. It's a lot more concentrated. We have reabsorbed a lot of salts, electrolytes, and we have reabsorbed all the glucose and amino acids that we could. This waste fluid then gets dumped into the collecting duct. You can see the collecting ducts like a common drain pipe that collects waste from all the houses in the neighborhood. Except in this case where this pipe is the collecting duct and the houses are individual nephrons. The collecting duct is important functionally from two aspects. First, it dips back into the medulla, and second, because antidiuretic hormone acts on our collecting ducts to decide whether our urine should be concentrated or dilute. The function of antidiuretic hormone is a whole different topic that we will discuss in some future video. The urine in each collecting duct is drained into the renal pelvis, which is this dilated portion of the kidney that is in turn connected to the ureters. The ureters connect the kidney to the urinary bladder that is connected to the urethra. Now to learn more about the urethra and all the other organs of human excretion, watch this upcoming video on our channel about the human excretory system. I hope you liked the video, and if you did, kindly support us by linking this video and sharing with your friends. Thank you so much for watching. Subscribe to support our channel.